Hello everyone, welcome to Life Science with Veena. We were talking about a colorimeter. Now we are going to see its part 2, that is the concept of complementary colors, how does the selection of filter occurs in the colorimetry and the applications of the colorimeter. Okay, so what is complementary color in case of colorimeter? So complementary color is the color which is opposite to the given color on the color wheel, that is called as complementary color. Now for example, the color of for example, this particular color is appearing red, right? But the color which it is absorbing, it is different than the red color. So there are two colors. One is this color of the solution, and the other is the color which is absorbed by the solution. So color which is absorbed by the solution is called as complementary color of that solution. So therefore, the filter which is used in the colorimeter should be of the complementary color of the solution. So now, why the complementary color is used in colorimeter? Because the maximum absorption of that solution can maximum absorb the light maximally at that particular wavelength, that is its maximum wavelength. So at the color of the complementary color, the light will be absorbed in maximum proportion by that colored substances. That is why the complementary color is used. So it will give you the high absorbance, and it will increase the precision, accuracy and the sensitivity of the substance, and will help to applicate the Beer-Lambert's law properly. That is why the Beer in colorimetry, the concept of complementary color is very important. Here you can see the color wheel is there, and here is the color wheel which has been fitted into a colorimeter. And here you can you set the wavelength by rotating this knob in this manner. Okay, so this is how it looks like. So the complementary of yellow is blue, the complementary of purple is green, the bluish green is red, the orange is blue. So these are complementary colors. And the complementary, so for example, if your solution is of yellow color, the filter of violet will be selected by you. Whereas if your solution is of red color, the filter for green will be selected for you. That is called as complementary color. Here you can see the approximate wavelength is given. This is the color which is absorbed, that is a complementary color, and this is the color of the solution. For example, for UV rays, it is not visible, so it will be below 400. It cannot be detected by colorimeter. You will need a spectrophotometer for that purpose. But if your color substance is of green yellow color, then you can go for a violet color as a filter. You can select the filter of violet color, which is at the 400 to 420 nanometer. Similarly, if your solution is yellow colored, you can select blue, because its complementary color is blue, which will be 420 to 500 nanometer. Red, the complementary is green. Okay, so this is the color of the solution, and this is the filter which can be selected, and this is the wavelength at which the filter is occurring. Okay, filter will give that particular color. Now let us move on to the applications of colorimeter. It is widely used in hospitals and laboratories, path labs, etc. for measuring the protein concentration, serum concentration, CSF, that is cerebrospinal concentration, cholesterol concentration, triglyceride measurements, glucose estimation, and various biochemical compound concentration you can determine with the help of colorimeter. So diagnostic purpose, it is used for the diagnostic purpose in the biochemistry labs, path labs, etc. They are used by food industry as well as manufacturers of paints and textiles for quality control. They are used to test the water quality by screening the turbidity, chloride, zinc, fluoride, cyanide, etc. colors. They can also determine the plant secondary metabolite concentrations or concentration of nutrients like nitrate, ammonia, or hemoglobin in the blood, and they can also identify the standard counterfeit drugs concentration, etc. So it has very wider application in botany, it has wider application in biochemistry, physiology, etc. Now, but there are some advantages and disadvantage, because the advantage is it is very inexpensive, easy to carry, easily transportable. You can carry from one place to another place, very handy, very well applicable for the quantitative analysis of colored substances. So these are some advantages. If you look at the disadvantage, you cannot go below 400 nanometer. That is, it is not applicable for the colorless compounds. You cannot go for a UV range or a micro or infrared. You can only work in the visible range. It will not work in the UV and infrared regions. That is a disadvantage. We cannot set a specific wavelength, as we have to set a range as a parameter. So 400 to 420. We cannot set 410 or 415 or so. So you have to work on the range, not this particular wavelength. Okay. Similar colors from interfering substances can produce errors in the result. So for example, the shades of the color are there. So it may hamper the accuracy or the precision and may lead to error. So these are some disadvantages of colorimeter. So this is how it works. Okay. So while using it, we have to take some preventions, like you have to read the user manual very carefully, avoid the bubbling while putting the sample. We have to bring the filter into place before switching on the colorimeter. We have to make sure that the cuvette is clean and dry. There are no finger marks, fingerprints or scratches on the cuvette. And before reading the absorbance, check that there are no bubbles into the solution. Remove the cuvette when the instrument is not in use. So these are some precautions that we have to use. Okay, so this is all about the colorimeter, concept of complementary colors, the application and the what you say, the application and the preventive measures and use, care and use. Okay. So this I have already explained. We have to keep it in a dry, cool and dry place. We have to take care. We have to do time to time servicing, and at the end of the day, we have to disconnect from the main switch and turn off the light lamp. And you cannot turn it on for the longer time, otherwise the tungsten lamp may get deformed, or it may become non-functional because of over usage or continuous usage. This is all about the colorimeter. So for more such videos, stay tuned to Life Science with Veena, and like, share, subscribe to my channel. Thanks for watching. Bye bye.